We are going to be going over cell membranes and cell transport in today's lecture. Oops, I need to shrink myself down first. Eep. I'm still here, just very tiny. All right, so the functions of the plasma membrane. What's the purpose of this thing? And it defines the outer border of all the cells and organelles in your body. And it manages what enters and exits the cell, receives different signals that initiate cellular responses, and it helps with joining to neighboring cells for different functions. We've talked about the plasma membrane, or the cell membrane, or the phospholipid bilayer. There's the different names by which we call it. And it's mainly composed of these hydrophilic phosphate heads, phosphate heads, and these hydrophobic fatty acid tails. Okay, you don't need to know which type of fatty acids, you don't need to know the composition of the phosphate head itself. You need to know that the tails are hydrophobic, meaning that they don't like water and they're made of fatty acids, and the hydrophilic heads like water and are made of phosphates. So we call this the phospholipid bilayer because, again, if you look really closely at this diagram, okay, you can see one layer and you can see two layers. Okay, on the outside are these phosphate heads, and these love water. So these are going to be on the inside and the outside of the cell. The other thing that you're going to see in this photo are all of these blue chunks in the cell membrane, and these are proteins. Okay, so they're integral or inside the membrane proteins. And these proteins are actually channels that we're going to talk about in a moment, about how stuff moves in and out of these channels when it's too big to just pass through the membrane. Now you have these different things that are attached to the top of these proteins. These are glycoproteins or glycolipids, and they have carbohydrates attached. These things can be used in a number of different ways. Often they are used with the immune system. They identify cells as self so that the immune system does not attack them. And they're receptors for many other types of molecules in the body. So those different types of receptor proteins, an example, like I was talking about, is the immune system, where these glycoproteins help to, they're called CD4 proteins. Our T cells have these CD4 proteins that recognize HIV as a self molecule, which is not a good thing. So this is an example of where these receptor proteins don't really help us out, but they do help they help in other ways with other diseases, just not HIV. So transport itself, the membrane, as I've said before, is selectively permeable, which means that only some things can pass through it without effort, if you will. So all cells like to maintain a balance of molecules between the inside and the outside, especially sodium and potassium. And so there are going to be different ways of transport for different types of ions or molecules like glucose or steroids and steroid hormones. We have two different types of transport. One of those is passive, that does not require energy, and the other is active, that requires energy, or ATP. So passive transport is the simplest type of transport, and an example of that is diffusion. So diffusion occurs when substances move from an area of high concentration to an area of low concentration, what we call down the concentration gradient. So let's say you have you're walking, you have a hill and then a valley, okay? How diffusion works is we have a high concentration here, and it moves down the hill to where there's less concentration, okay? So this is how diffusion works. So if you spray perfume on one side of a room, eventually, over a period of time, you're going to smell that perfume everywhere in the room because it will have diffused from an area of high concentration where you sprayed it to an area of low concentration, the other side of the room. The movement of these molecules will actually decrease or stop once the molecule has spread out into space and is evenly spaced out. We call this equilibrium. Okay? So we're always trying to achieve equilibrium. Now, only certain molecules can diffuse. Okay, things like oxygen, CO2, steroid hormones, okay, those can diffuse right through the lipid bilayer in and out of the cell without energy. There are different types of factors that can affect diffusion, how fast something spreads out. So concentration gradients are one. Oops, I don't want to do that. Concentration gradients are one example, where the greater the difference you have, the faster the diffusion. So the more concentrated something is in one area, the faster it's going to diffuse to another area. Think of your hill as being very steep there. The mass of the molecules, how heavy they are. So smaller molecules are going to move faster. Temperature, molecules always move faster when it's hotter out. That's why your body moves easier when it's hot, and when it's really cold, your body moves really slow because all of the molecules in your body are also moving very slow. The density of your solvent, we're not going to talk about that in detail. Solubility, so nonpolar or lipid-soluble molecules are going to diffuse faster than non-lipid molecules. Smaller or increased surface area will actually speed up diffusion rates. Distance traveled, the greater the distance that something has to travel to spread out, the more time it's going to take. And then pressure. So in some cells like kidney cells or in plant cells like we'll talk about in a minute, pressure forces solutions through membranes, and this can actually speed up diffusion rates. So another means of transport using these protein channels that I talked about earlier is facilitated diffusion. You can call it facilitated passive transport. I like facilitated diffusion. Now, this moves things down, substances or molecules down their concentration gradient still. So we're still going from an area of really high concentration to an area of low concentration. But these molecules are too big or polar and can't get through the cell membrane by themselves, so they have to go through these special channels to help them into the cell. So we're going to go, we're in a special channel there. Okay, osmosis is the diffusion of water across a membrane. Water is always going to move from an area of high concentration to an area of low concentration. That's what we're going to talk about with osmosis. Tonicity is, there was something I wanted to talk about back here. Oh, well, I will review in a moment. Oops, I'm going the wrong way. Tonicity. Okay, so tonicity is, again, we've talked a little bit about isotonic, hypotonic, and hypertonic solutions, but a hypertonic solution, okay, hyper is above, has lower osmolarity than the cytosol. So water is going to leave the cell. Okay, hypertonic, there's more water outside the cell. So the water is going to leave the cells to go outside. In an isotonic solution, there is equal amounts of water or osmolarity inside and outside of the cell, so water doesn't move. In a hypotonic, hypotonic, water is going to enter the cell. Here we go. So this should say hypo. My apologies. Oh, hypotonic. So in a hypotonic solution, okay, extracellular fluids have higher osmolarity than the cytosol or than inside the cell. So water will enter the cell. So what that means, again, hypotonic, meaning that there is less water inside the cell and more water outside, so it will go into the cell. A really good example of this is plant cells, because plant cells, the cells themselves will actually pull away from the cell walls depending on the tonicity of the solution and the turgor pressure inside of the plant. So PM means plasma membrane, CW means cell wall. Okay, so cell wall, CW. So organisms who have cell walls, plants, fungi, bacteria, some different protists, are going to actually prefer this as opposed to this, which animals prefer. Sorry, my writing is so terrible. So our hypotonic solutions are what plants are going to prefer because that gives them the most water in their cells and allows them to stand up the straightest. So when you have hypertonic solutions in plants, the plasma membrane actually shrinks and pulls away from the cell wall, which really isn't good for the plants. All right, active transport is any time that an ion or molecule that needs to be transported through a membrane like this. So maybe we have a few molecules, and there's a lot of molecules inside the cell, but we still need these molecules to go in. So now we are going from a low concentration to a high concentration gradient, okay? And that's when we need to use active transport, and that's when we need to use energy. So energy is always required. Our energy is going to be ATP. That's what we're going to talk about. So a little bit of a review, before we get into the bulk transport, okay? So diffusion, picture a doorway, okay? Picture a doorway. And diffusion is when you're just moving in and out of the doorway. One side of the doorway is inside the cell, one side of the doorway is outside of the cell, and the door and the wall are the cell membrane. Okay, so you're moving in, you're moving out. We'll use this example here. Okay, here's the door. Okay, diffusion, I'm going to move in the door, I'm going to move out the door, and it's easy peasy. Oh, stop it. No one's here. It's just mommy. Okay, so that's diffusion. So Lola, stop that. So facilitated diffusion, I want you to picture like a shopping mall. And there are always these little sort of entrance chambers when you go into a shopping mall. So you enter through opening automatic doors. You enter the little entrance chamber where there's normally, depending on the weather, either a lot of AC or a lot of heat blowing on you, and then the automatic doors open again, and then you enter the store. I want you to think about that as facilitated diffusion. So you have your doorway here, you're at the doorway, automatic doors open, you step through the tube, automatic doors open, you're inside the cell. You don't have to expend energy, but you go through a special tube to get in. Active transport, you actually have to expend energy, open the door, expend energy to get into the cell, okay? So diffusion, no energy, back and forth. Facilitated diffusion through protein channels. Active transport uses ATP. There's different types of bulk transport. There's phagocytosis, pinocytosis, and receptor-mediated endocytosis. In addition to exocytosis. We're going to use our roots here. Okay. Endo means inside. Exo means outside. So endo and exocytosis at their most basic are moving substances inside and outside the cell. They do this, and phagocytosis is doing the same thing, but phagocytosis, this means eat or food. So phagocytosis itself is actually cell eating. Okay, and that's basically when a large particle is engulfed by a piece of the cell membrane, and a piece of the cell membrane buds off into the cell containing the particle in a vacuole. Exocytosis, Okay, we're going to have a vesicle of membrane, and it is going to attach to the cell membrane and open up and allow its contents outside of the cell. And endocytosis does the opposite. In endocytosis, we have a vesicle and we kind of bud into the cell. Okay, so exo out, endo in, phago eat. All right, that is, you want to keep our annotations? We're going to discard those. Our cell transport lecture and enzymes, I believe. If I check my, yes, enzymes are up next. See you in the enzyme lecture.